Albugo or cystopus. It is one type of fungi, Albugo or cystopus. Now we discuss about the classification of Albugo or cystopus. Division Eumycota, subdivision Mastigomycotina, class Oomycetes, order Perinospores, family Albuginaceae. Division Eumycota means true fungi. Subdivision Mastigomycotina means that fungi produced spores with flagella. Class Oomycetes, order Perinospores, family Albuginaceae. Now discussed about the Albugo white rust disease. The white rust disease caused by Albugo fungi in Cruciferaceae or Brassicaceae family plants. When we observed the white rust disease on the plant leaves, the white color patches or spots clearly identified. The white color rust-like structures are developed on the dorsal side or ventral side of the Cruciferaceae or Brassicaceae leaves. Now, these fungi containing thallus structure, thallus means the fungal hyphae. The fungal hyphae contain tube-like, branched, filamentous-like structure. It contains a thick cell wall. The cell wall mainly made with cellulose, hemicellulose, and chitin chemicals. Below the cell wall, a thin plasma layer is present. It made with phospholipids and protein chemicals. Below the plasma layer, cytoplasm is present. In that cytoplasm, more number of diploid nuclei are present. The diploid nuclei are present in the cytoplasm. Here it is the more number of diploid nuclei present in the filamentous. This structure is called cenocytic or multinucleated structure. Mainly the fungi of Albugo received food material, one bird-like structures they called Historiums. Historiums are mainly helps in collecting of food material from the host cells. It is the mycelium or thallus structure of Albugo. When the mycelium is contaminated on the Cruciferaceae or Brassicaceae leaves with the spores, the spores are landing on the leaves. They are absorbed moisture from that leaves. That they are produced to germitube. The germitube enter into the mesophyll through the stoma, and they are enter into the mesophyll. They occupied intercellular spaces. For the development, the mycelium or germitube collected food material from the host cell through the nodular-like structures they called Historiums. It is the fungal hyphae structure. Now we discussed about asexual reproduction. The mycelium participated in asexual reproduction when the favorable conditions only. Around the Albugo mycelium, if favorable conditions are present, that favorable conditions are mainly helps in the asexual reproduction of fungi. See, it is the sexual reproduction of fungi. So, diploid spores are landing on the dorsal side of leaf surface. They absorbed moisture on the leaf surface. They develop into germitube-like structure. The germitube enter into the mesophyll through the stoma. It is occupied intercellular spaces of the host leaf. The mycelium need food material. The needed food material collected from the host cells through the Historiums. When the favorable condition, the mycelium produced aerial stalk-like structure on the dorsal side of the host leaf. See, here it is the aerial stalk-like structures look like a club-shaped like structures. The club-shaped structures are called Sporangio Pores. Initially, the Sporangiopore terminal pod contains more number of nuclei, diploid nuclei with cytoplasm. Around these more number of diploid nuclei, cell wall is formed. It converts into sporangia. Here it is the same way. By the basipetal succession, more number of sporangiums are developed from the each sporangiopore. Here it is the first developed sporangium far from the sporangiopore. and early developed sporangiopore near the or above the sporangiopore. Finally, all the sporangiopores are produced more number of sporangiums. The more number of sporangiums are white in color. They are pressure on the upper epidermis. By this pressure, upper epidermis is broken. White color sporangiums are comes out from the host leaves. The white color spots are clearly identified during the asexual reproduction of the albugo. The sporangiums containing multinucleate structure, each nucleus develop into zoospore-like structures. The zoospore containing two unequal flagella. One is tin cell flagella and whiplash flagella. It is whiplash flagella, is tin cell flagella. The zoospore are in motile condition. The motile condition zoospores are containing diploid nucleus. When the germination time, the zoospore loses their flagella and encrusted zoospores to form encrusted zoospores. That encrusted zoospores are develop into germitube-like structure. See the development of zoospores when in environment more than 20 degrees, less than 20 degrees of temperature, less than 20 degrees of temperature present in the environment, the nuclei, the diploid nucleus develop into zoospores. Less than 20 degrees Celsius of temperature present in the environment, the diploid nucleus develop into zoospores. Sometimes the temperature will be increased. If the temperature will more than 20 degrees Celsius, the diploid nuclei directly develop into spores-like structures. The spores are called aplanospores. The aplanospores are moved by wind. They are landing on the healthy cruciferous leaves. It is the continuation life cycle. So the development of sporangium is based on the temperature. If below 20 degrees Celsius temperature is present in the environment, they produce zoospores. Above 20 degrees Celsius temperature present in the environment, they produce direct aplanospores. Finally, the two spores are absorbed moisture. They are developed into germitube and landing, develop into germitube. They are enter into the mesophyll through the host cells. It is the continuation life cycle in asexual reproduction. Now, we discussed the sexual reproduction. The sexual reproduction by the conjugation mechanism or touch mechanism or contact mechanism. The sexual reproduction of albugo in unfavorable conditions. Around the albugo mycelium, we observed unfavorable conditions that myceliums are participated in sexual reproduction. See, during the sexual reproduction in unfavorable condition, first two reproductive organs are developed. One is large in size, it is in club shaped called oogonium. It is the female reproductive organ. The female reproductive organ containing club-shaped structure. In this oogonium, more number of diploid nuclei are present. Lateral side of this oogonium, male reproductive organ is developed. The male reproductive organ is called antheridium. Antheridium also club-shaped structure, but is small in size. It containing more number of diploid nuclei. In Albugo blighty, see here it is, in Albugo, two major species are present. One is Albugo blighty and second one is Albugo candida. In Albugo blighty, more number of diploid nuclei are present in the oogonium. More number of diploid nuclei are present in the antheridium. These all diploid nuclei of antheridium and oogonium are participated in the meiotic division. They produced more number of haploid nuclei. Up to 100 haploid nuclei are developed in the oogonium. Up to 100 nuclei are developed in the antheridium. When the matured stage, the antheridium contact with the oogonium. By the contact position, the cell wall is lysed between the antheridium and oogonium to form a one conjugation tube-like structure. See, the conjugation tube-like structure is called fertilization tube. By the fertilization tube, more number of male nuclei from the antheridium enter into the oogonium. Before the development of oogonium, the nuclei or cytoplasm divided into two major portions. One is periplasm and centroplasm or nucleoplasm. Periplasm containing only liquid cytoplasm It present at the peripheral region of the oogonium, and at central region of oogonium, nucleoplasm is present. The nucleoplasm is called oospore. The nucleoplasm containing more number of haploid nuclei in Albugo blighty. See here it is when the conjugation or fertilization tube formed between the antheridium and oogonium. From the antheridium, more number of male haploid nuclei are enter into the nucleoplasm of the oogonium. They are fused with the oogonium haploid nuclei to form diploid zygotes. See here it is only nuclei are fused. This process is called karyogamy. After karyogamy, plasm, cytoplasm of antheridium, cytoplasm of oogonium are also fused, called plasmogamy. Plasmogamy after karyogamy is completed to form diploid zygotes in the oogonium. The diploid zygotes are participated in the meiotic division. Diploid zygotes are participated in the mitotic divisions, not meiotic division. They participated in the mitotic division. They develop into diploid zoospores when in the environment temperature is below 20 degrees Celsius. The zoospores are developed into germ tube in the cold temperature time. If in an environment more than 20 degrees Celsius of temperature is present, the zoospores cannot develop. Direct diploid nuclei are developed into aplanospores like structure. It is in Albugo blighty. But in Albugo candida, haploid, only from oogonium, and antheridium, one haploid nuclei and one haploid nuclei are developed. From antheridium also one haploid nuclei, one oogonium also haploid nuclei are developed. These two nuclei are fused to form zygote. Zygote participated in the mitotic divisions. They develop into zoospores. When the cold conditions, they are directly developed into aplanospores in the dry conditions. It is the continuation life cycle also in the sexual reproduction of Albugo. Sexual reproduction of Albugo. Now we observe the life cycle. Here it is always we observed in the life cycle of Albugo, we find all stages with diploid conditions. Here is Albugo blighty containing diploid nucleus. The diploid nucleus participated in the asexual reproduction by the sporangiums. They produced again diploid Albugo mycelium. If they are participated in the sexual reproduction, they can produce the sexual reproductive organs like oogonium and antheridium. Oogonium and antheridium are both in diploid condition. The nuclei of antheridium and oogonium participated in the meiosis to produce male nucleus and female nucleus or oospores. These are fused, fertilized. These are fused means fertilized to form oospore or zygote. The zygote germinated into zoospores. They are germinated into diploid Albugo mycelium. When we observed in the total life cycle of Albugo, haploid stages is the minute phase, temporary phase, but remaining phases of mycelium containing diploid nucleus. So these life cycles are called diploidic life cycle or diploid life cycle. It is the Albugo life cycle.